All right, I think we'll go ahead and get started. So we're in the home stretch here for the morning. And I wanted to sort of, again, move up to this final scale of imaging tissue. And so I'm going to present basically an overview of some of the different imaging technologies that we do in my own research group here at Beckman, and apply them sort of at the tissue level. But you'll see that everything is kind of connected. So we're also looking at cells, and we're looking at the roles that molecules play at all these different scales. I showed this slide in my welcoming introduction, but it's changed a bit because what I want to emphasize is in biomedical imaging, we primarily are used to these types of modalities at the clinical end, and they're very good for imaging at the more the tissue, the organ, the body level, both for structure and function. But again, it's this biophotonics that brings us down to this scale, and we start looking at technologies like OCT, pathology, fluorescence microscopy, some of the other imaging modalities that we heard about from Professor Kong and Myung this morning. So to give you perspective what goes on in my own research lab here, and partly because, as we'll talk about, or you'll hear, there's going to be tours and demos and hands-on labs for a couple of your afternoons here. And so one of those is actually in my own lab for looking at OCT. But in my lab, what we do is we do a wide range of things, ranging from developing hardware, including novel optical sources that we build and use for different types of microscopes or portable systems. And the goal is really to develop a lot of technologies for translating these biophotonic technologies into clinical applications. And that hardware is coupled with software or computational approaches to extract information from the optical signals, to improve the image quality, the resolution, and many different computational approaches. And then those are applied at many different scales. So beginning at the cellular level, looking at developing engineered tissues, and much of what I'll show today is going to be at the kind of the cell, the tissue level. We do a lot of optical coherence tomography for structural and functional imaging, and you'll see that in my presentation as well as optical elastography. So being able to use the optical signals that we measure and understand how the tissue may deform or move. And that can provide us information about the biomechanical properties of tissue. And then finally, for us to get to the molecular scale, we talk about not necessarily imaging molecules, but using contrast agents to target specific molecules and understand that molecular distribution. We started looking a bit more into neurophotonics, and I won't talk about that today, but it's really, I think, a growing and interesting area how light can interact with neural systems. But much of what we do and develop in my group ultimately is targeted for these translational applications. Things that the use of light, because it's relatively low power, safe, readily transported, relatively low cost, we can transition and do studies in humans quite readily. So we've got in my lab a number of studies looking at primary care applications to identify ear biofilms and infections. And something I'll talk about next week is our intraoperative use. So using OCT and other techniques to look at breast tumor margins and lymph nodes, and we'll focus on that. But today at the tissue level, I chose a particular tissue that tends to be a favorite for a lot of optical imaging technologies, and probably because it's accessible, but it's also incredibly dynamic. So of course it's not just hands, but it's skin, and skin really, I think, is a remarkable organ, but just because of the multiple structures that are present, just the dynamics that can take place. There's obviously pathologies with cancer, but even dermatitis and acne and everything that we can all relate to. So there's a lot of targets at this tissue that we can use imaging to interrogate. And because it's so optically accessible, it is kind of a favorite for us in biophotonics. So what I want to talk about today is to walk through a series of different types of optical imaging modalities that we've combined and used in different ways to interrogate skin and other tissues. To use some of these techniques to look at in vivo mouse skin and then human skin, talk a few slides about his elastography, and then just kind of where we're going with all of this. So to start, this is perhaps more at the cellular level as we transition, but we're interested in using imaging techniques to do intravital microscopy. So being able to understand and image the cell dynamics that take place dynamically over in living specimens or even in humans. And this has really been an indispensable tool for cell biology, but there's really some fundamental limitations. One of which is that there's limited contrast, because if we're looking at trying to understand the cell dynamics or these in vivo behaviors and interactions, you don't necessarily want to add contrast agents all the time. These often can limit the viability of cells or disrupt their natural behavior. And particularly if we're looking long term, this is difficult. The second is that limited time window. So, this is a video clip looking at some of the cells in a mouse skin over about 60 to 90 minutes. And usually we can, keep an animal down for an hour or two. But anything longer than that requires some new ideas, and we'll show you some of our solutions to that. But we want to be able to extend those time windows to days, to weeks, months, where a lot of the processes in biology may take place over those time spans. So some of the modalities I'm going to emphasize today, the first three are here. OCT, and I'll talk about a little bit about each of these, which is going to give us more about scattering intensity or tissue structure. Again, label-free. And it also, because we can look at the phase difference in the light signal we recover, we can get maps or images of the vasculature moving red blood cells through the capillary network. MPM is multi-photon microscopy, and that's really a collection of nonlinear techniques, such as two-photon excited fluorescence, that's going to be used to look at both autofluorescence, which tells us something about NAD and FAD and other autofluorescent molecules in tissue and cells, or in this case we can use GFP or other genetically expressed markers that we can identify and track. Another multi-photon approach is second harmonic generation, and that's very good at looking at collagen structure. And of course the skin and these extracellular matrices that Professor Kong was talking about are filled with collagen. And we can use that nonlinear technique to be able to look at the organization, the density, and the structure. FLIM is fluorescence lifetime imaging microscopy. And that really tells us something about the metabolic functions that are taking place in the cells, or the viability of the cells, or the molecular dynamics. And combining all these into a system really gives us a more comprehensive view of what's going on within our tissues. So just to give a little bit more detail of each of these, because I'm not sure really if everybody's heard about this, but OCT is a technique that was developed about 25 years ago now, but it's essentially the optical analog to ultrasound, and we collect reflections of near-infrared light. We use interferometry to localize where those reflections came from, and we can get things such as 3D volumes. These are individual cells within the respiratory tract of a tadpole. We can get very fast temporal acquisitions such as the beating heart of that tadpole, and all label-free, just based on the inherent optical scattering properties of the tissue. Another technique, well, just to compare that, you've probably all heard of confocal microscopy. Confocal microscopy uses a pair of pinholes, so you have a point source that's then focused into tissue to a focal plane to excite those fluorophores. And you have a pinhole in front of a detector that's placed confocally with the source. And so only fluorescence that's emitting from that focal plane is going to be passing through that pinhole and get detected. And this is really a spatial rejection of out-of-focus light. And it works quite well, but its depths are quite limited to maybe tens of microns in highly scattering tissue. OCT, on the other hand, does this spatial rejection. We often use pinhole fiber optics, single mode optical fibers that have a very small 9-micron core or so that act as a pinhole. But in addition, any light that may get bounced around in the tissue that happens to get collected back into that optical fiber is also, those are multiply scattered photons. Those will get rejected just because they're no longer coherent with the light, say, in the reference arm of an OCT system. So OCT gives us both spatial and coherent rejection of that out of plane light, or that multiply scattered photons. And because of that, that's what gives OCT the property of being able to image deep, up to perhaps several millimeters in tissue, depending on the optical scattering properties of the tissue itself. Now, multiphoton microscopy, I'd mentioned 2-photon excitation. That's, in comparison to 1 photon, where we put in optical energy, there's an absorption event, there's a non-radiative decay, and then there's a fallback where you get this fluorescence or this emission. We can achieve that same effect with 2 photons that are longer in wavelength, but half the energy, that still allow that same type of excitation and then emission. The advantages of this, of course, that we're using longer wavelengths for excitation. That's generally safer. It's less absorbed in biological tissue. It gives us deeper penetration, less autofluorescence. The fact that this 2-photon event has to occur both temporally and spatially in a well-defined manner gives us this higher resolution. So this is a classic example from Cornell, that here you have a microscope objective on the right that's focusing light into I think a cuvette of rhodamine dye, fluorescent dye, and this is 1-photon excitation, and you can really see the beam profile that's exciting those fluorophores. Coming in from the left is 2-photon excitation, and you can see the small little focal spot of fluorescence that's produced. And so we no longer, in the case of 2-photon microscopy, we no longer necessarily have to rely on this type of gating, spatial gating, because we're only going to get signal from a very small, well-defined, spatially and temporally localized area. Another 2-photon process, which we'll talk about, is second harmonic generation, which is essentially the same type of 2-photon excitation, but it's to a virtual state, and we get this emission, this SHG emission, and this is typically will come from structure or ordered features, molecules in the tissue, and collagen being one of the more dominant ones. So we'll talk about this. The disadvantages of these, of course, is that the systems that are used for nonlinear optics tend to be much more complex. The lasers are much more expensive. Ultimately, any nonlinear signal is inherently weaker, so we have to deal with weaker signals and trying to measure those with more sensitive systems, detective systems. But the payoff, you'll see, is, I think, is worth it. FLIM now, very briefly. Just as we talked about that fluorescence emission, there really is a time constant that's associated with that emission. And if we can measure that emission over time, we can look at those time constants. That is a very sensitive metric that depends on the molecular changes in that microenvironment, pH, ion concentration, again, metabolic activity. These types of things will change that fluorescence lifetime. And so we have systems that will measure that, and it's a way of telling us, again, about the metabolic or viability state of these cells that we're imaging. Well, how do we put this all together? One of the things that we focused on in my lab is to integrate these modalities, because oftentimes they'll use just a single source, a single laser source, and yet each of these has very different contrast mechanisms based on different physics. And if we can spatially, and you'll see also maybe temporally, co-register all this image data, then it opens up this idea, can we, in a combinatorial fashion, try to understand, in any given voxel, what is the optical, properties of that molecule or tissue in that voxel of light? And can we even define sort of an optical basis for disease and define, a very unique definition of what's going on when we have all this data.
Yeah. When you do the image reconstruction in this case, do you treat the three modalities separately and then combine them later, or is the image reconstruction somehow fusing the data together?
Yeah. So we do both. So the physical acquisition, as we'll see in this case, maybe I can show you, we have our optical source, but we have different detector arms. So we have here's our sample here. So that's going to obviously produce fluorescence or backscattered light. Our fluorescence is picked off and we can send some to our FLIM. We have multiple PMTs for that SHG signal or other signals. Our OCT signal comes back here to an interferometer to collect that scattering or phase information. So we have different detectors. Those are all collected at the same time. And so we have separate data sets. But one of the things that we're doing right now is to now look at this more mathematically and can we combine them? Is the sum greater than the parts? And can we have different weighting? Can we, another way, if people are familiar with linear algebra, is to think about if we have all these different parameters, can each set define kind of a unique eigenstate or a basis function that represents, the state of the system? And that's what we're headed toward. So the other parts of this system you can see is the laser that we actually will do two things. We'll split that output and send one to a photonic crystal fiber, which spectrally broadens our light for OCT. The other is just to send this directly so we have kind of high peak power for exciting our fluorophores for nonlinear optics. And we combine these two spectra. So what we get is what we call a dual spectrum source. These are the two different spectra, and then when they're combined, on a spectrometer, our light output kind of looks like this. But we can tune this. We have a tunable Ti-sapphire. So as we tune our center wavelength, we can both preserve that broadening, but also that central peak, and this sort of tunes. And we can then use that to selectively excite, different molecular species. In this case, these are fibroblasts that express green fluorescent protein with their adhesion receptors vinculin. It's also co-stained with rhodamine and a nuclear dye. So we can then, at different wavelengths, optimally excite the nuclear dye here, or the rhodamine in the cytoplasm, or the GFP that's on the cell membrane of this fibroblast. So this gives us a lot of flexibility of the source and excitation. Okay, well, our target being skin, we've had many different projects looking at skin, both in mice and in humans. We've also engineered skin and look at how this engineered tissue engrafts onto hosts and how that integrates. And some of that work, in fact, was done with Professor Kong as a collaborator. We've also been interested in stem cells in skin, and what we are wanting to do is track where these stem cells come from, where do they reside, where they go in different wound healing or different pathologies. And so the hypotheses that exist in the field is that stem cells come from our bone marrow, will go to this bulge area, which is within every single hair follicle. And when you injure your skin or have a wound, then these stem cells may migrate out of this bulge and repair the wound. And so these types of dynamics is always an open question, and that's where we've been trying to apply some of these techniques. So you can see that there's many different questions that we can try to answer with these optical techniques. Well, here's just some examples. So we're starting with mouse skin. This is multiphoton, primarily SHG, that what we're looking at is the collagen structures at different depths. And these are individual hair follicles. And we actually have some videos that shows the co-registered multiphoton and the OCM, or OCT. And so you can see at different depths there's different structures, obviously different contrast mechanisms. OCM is giving us all the backscattered light, whereas MPM is really just showing us the collagen. And we can see the hair follicle here, and again, different contrast, different physics for generating that. The the data we can then assemble into 3D volumes, and we can look at kind of the interesting orientation of the collagen fibers in 3D as they form around these follicles. You can see that there's differences in resolution that we have to always account for between the different modalities. But again, like I said, different contrast and different physics. Well, some of the central applications that we want to use this technology for is to look at wound healing, and also this recognition that wound healing takes place over years. So you wound yourself, immediately within minutes there's coagulation events, there's vasoconstriction, followed by vasodilation for bringing in an inflammatory response where immune cells and stem cells enter into the area. And then there's fibroplasia, there's granulation tissue, so scar formation starts to set in, collagen reorganizes, we get contraction, we get angiogenesis. But then it could really take years after that for the collagen to remodel. And even after it remodels, it's only about 50% as strong as that original collagen matrix. And so these are some of the things that we don't really understand why that doesn't completely remodel under these conditions, but these are questions we wanted to ask. Well, the main point for our studies is that this takes place over, again, months and years. And so if we have time-lapse imaging that only occurs over minutes to maybe an hour or so, we're really missing a lot of these events. And so we've had to solve that problem. So let me set up the model that we're using, and then I'll talk about some of our solutions. Well, the model we wanted to use that we developed was to take bone marrow from a green fluorescent protein expressing mouse and do a bone marrow transplant. So we extract the bone marrow from this GFP mouse and we transplant it into wild type mice that have been immunodepleted. So these mice have been exposed to radiation that basically kills off any of their normal bone marrow. And these mice will die unless they get bone marrow to recover. And so we do that, and what we then have is a wild type mouse that only has GFP cells coming from the marrow. And if we see GFP cells anywhere else in the animal, we know they came from the marrow. And that could be stem cells, it could be immune cells, it could be hemopoietic cells, but the other advantage is that it gives us a limited cell population to study. So anytime you look at tissue, in general, with any technique that has cellular resolution, you see all these cells, and you don't know which cells are which. It's hard to figure out what their roles are. But if you somehow can label and uniquely identify your cell population, you can start to see interesting dynamics. So our system, again, has OCT, which gives us this structural information. What you're looking at here is a mouse ear, and this band is kind of the collagen band, but the overlying skin above that. We can use phase variance, which only gives us the varying flow or phase variance from the flow, and therefore map out that microvasculature. Our MPM is going to give us two channels, a GFP channel for the bone marrow derived GFP cells, and then an SHG channel for the collagen. And we combine all these together and we get images that look like this. Again, spatially co-registered. The color scale here, the gray is that OCT variance. And so what you see here are little dark spots. Each of those dark spots are a hair follicle. And interestingly, the capillary network nicely encircles each of those. And then the red channel is our SHG, so the underlying collagen, and the green is the GFP. And so we can look at that data in cross-section. We can see the SHG in the dermis of the skin. And if we look at where this wound was placed, we can see this migration of these GFP cells, again, from the bone marrow, coming to the site of the wound and accumulating there. Now, so just really a wealth of information that gives us spatial information. If you just use fluorescence I often say when people use just fluorescence microscopy, with fluorescence you only see what you're looking for because you have to label it or you have to understand where that fluorescence is coming from, but you're missing a lot of the other underlying structure and components unless you physically label all that. But with this combined approach, we start to see things in their spatial correlations that are rather interesting. This was just another example. I apologize, the color scale changed, but here now the red is the vasculature, the blue is the SHG, and the green is still the GFP. We can collect the cross-sectional data that shows this wound. We can see that the wound, we see the scattering from the OCT in this wound that's repairing. We also see the green cells around the periphery. We see the SHG from the collagen. You see the wound is void of collagen and any vasculature. But again, we can start to see that relationship. But
yeah, I don't know if I missed this, but what image you input?
Okay, so this was in mouse skin. It was just really a small wound, like a punch biopsy, and I don't have a scale bar, but these are typically just a few hundred microns across. So very small, almost just like a pinprick. But a small punch biopsy where we take out a small bit of the epidermis and dermis of that. And we also take out some of the vasculature with that, you'll see. Yeah, so that's the wounding that we're doing. This is either taking place on the ear of the mouse or on the back skin. So that's sort of a snapshot in time, but we really want that time-lapse imaging. A colleague of mine, Charles Lin at MGH, coined this term slow microscopy, and I always like that because everybody wants microscopy to be fast in real time and capture these dynamic events, but when you are looking at events over months and years, you want slow microscopy. You want to be able to capture this. So that's another way of thinking about this long-term effect. Well, what we want to do is if we can only bring a mouse back every a day for a couple hours, then we can always collect data every day for that time period. But then it becomes a problem, how do you spatially co-register all that information? And people have done this, but a lot of people use what's called a rigid registration. So you take your image matrix from day 0 and day 1, and you maybe rotate or you translate or you maybe scale different axes to try to get those to overlap. But that doesn't work so well if the tissue is deformable, as is in the case of skin. And so then you have to go to a more of a non-rigid registration where you have this perhaps morphine or this deformation matrix that relates the two different days. And that's what had to be done for skin imaging. Well, if that's what we're going to use, then we still need landmarks. And fortunately, our SHG channel provides us some really nice landmarks of these follicles. And these follicles don't change over months or perhaps even years. And so we have an image from day 1 and day 2, but then there's the question of which follicle is which. And so we have this follicle on day 1, but which of that is on day 2? But you can look at not just the single follicles, but then the patterns of the follicles, and look at its neighbor and say, for this one, what do its neighbors look like, and then try to match the regional pattern of those follicles, to a deformed pattern on the second day. Okay, well, we can automate, algorithms to do this, and that's what Ben Graff, who is a PhD student in my group, really worked on this for his thesis, and he developed this algorithm then that would automatically search and identify those corresponding follicles between two different days. And this is day 0, the normal skin. This is day 1 after there's been a small wound that's placed. And so it's an iterative transformation. It sort of checks to see if it makes sense and comes up with the best match between the two different days. And these are the results. So if we have flashing between day 0 and day 1, with and without, or without and with a wound, if this is unregistered, you can see that that follicle is moving back and forth. So that matrix hasn't been registered. But if we do register that using this approach, then every one of these follicles is now in the same location. The wound is what's doing the distorting of that tissue. And so when you do this, you end up having a matrix that relates the two, and that's something that can be used to look at, what is that non-rigid change between those two different days, and did the tissue expand or contract between those. So automatically we start to get some biomechanical information about the tissue as well. So like all good engineers, we want to validate this quantitatively. And so what Ben did was he took image 1 before the wound and labeled it red, and then 2 in green, and with the different types of registration, looked to see how well that matched. So if we look at making every follicle kind of open, and you try to register this, if you see red behind the green, well, that wasn't matched up. If we used, that's no registration. If we use the rigid registration, we still see maybe a couple follicles look open, others don't match. But if we look at the non-rigid registration, a lot of the follicles you can see right through. So we've really developed this nonlinear, non-rigid deformation matrix or grid between the two data sets. All right, well, let's put this all together. So we have 3D volumes of different imaging modalities at each time point, and then we have this transformation between each of these days. And this is just a sequence over 64 days of these different data sets. This is, again, a skin, a mouse skin with a wound, and you can just start by looking. Here's the OCM. You can see the original skin, the wound is placed, the scab is formed, and over time we start to see this healing of that wound. If we look at the GFP channel, again, the bone marrow-derived cells, we see the original skin, the wound, a scab. We see this rise and influx of these inflammatory cells and bone marrow-derived cells all around the skin from that wound. If we look at the vasculature, we see the original wound is void of vessels, and then over time we see angiogenesis as the vasculature is filling in that wound. And finally, the SHG channel, which again shows that collagen that's beginning to fill in and become organized and acquire that signal. So just a wealth of information that we can acquire with this type of system. Some interesting points about this are shown here. So if we look at the collagen and the structure from OCT, we can see that there's really different patterns of healing. And that's again because anytime you make a wound, there's going to be an influx of tissue and repair going on in the wound. But the collagen, the ordered arrangement of that collagen that will give the SHG signal occurs on a much longer time scale. So you can see the gray is the OCT that's filling in that wound, but it's taking a little bit longer for that collagen structure to organize itself. In addition, we can use that warped grid information to look at all these parameters, such as wound contracture. And so wound closure is this dark line which kind of dips and then plateaus out here. That collagen synthesis I told you about, it really takes a much longer path to sort of recover. The wound contraction is shown here, and then the angiogenesis is void for a few days and then comes up. So we can look at these trends over time. Yes?
Have you tried to increase the punch size so you can see the reduced healing and then how actually it influences the angiogenesis?
Yeah, we have looked at different punch biopsy sizes, up to even maybe 3 to 5 millimeters, and we do see very different events with each of those. And a lot of it is, we don't have definitive results, but a lot of the stem cells that may be coming from those hair follicles will go, will migrate from the hair follicles, will differentiate into keratinocytes to help repair that skin. And if that distance is so far, is so great, we see a lot more delayed type responses here. And so, yeah, that wound size really does matter. And I don't handle, but at some point we need that engineered skin in there to facilitate that process. So. Okay, and these are just some snapshots again over that time sequence to kind of show you those differences that are happening. How can we use this? Well, there's a lot of different applications. One of those is to look at tracking the immune response to nanoparticles. And so everybody should be using sunscreen in the summer. Sunscreen contains zinc oxide as well as others, but those are nanoparticles, and there's always a question of, what do these nanoparticles do? Are they safe? Do they get into our skin? What sort of response does our body have to these? Well, we use this system to look at that. And fortunately, the zinc oxide produces a 2-photon luminescence signal. So there is if you hit it with this titanium sapphire with a ultra short pulse, it will actually luminesce and produce its own signature. And so we can have a channel that's specific for zinc oxide. And then this is our SHG. This is our green fluorescent protein from our immune cells. And this is an example of a data set looked at over a couple of days of intact skin with a topical application of zinc oxide. And what we've seen, fortunately, is that with an intact stratum corneum, that zinc oxide doesn't get through. Nanoparticles don't go through. That our stratum corneum is really a remarkable barrier to things. And we don't see any immune response from that being on our skin. We had to look at a positive control to make sure our system was working. In this case, we did kind of two things. We made a small linear incision, just took a scalpel and made about a millimeter long incision, and then topically applied LPS, lipopolysaccharide, which is what bacteria produce. It's not bacteria directly, but it's very much an inflammatory agent. And so we can see this very rapid rise of these immune cells into that site. And we knew that was a strong positive control. Well, next then we took and did a topical application of nanoparticles now in the presence of a linear incision. And so our SHG, you can see that linear incision in the collagen. We can see that there's a slight increase in the GFP cells here. We can see evidence that indeed those nanoparticles are in the wound itself. And then we looked at a negative control, which is just saline inside of that linear incision, and we don't really see any significant change. So if we look at this more quantitatively and statistically, then we see, of course, that big LPS treatment has this significant change in recruitment of GFP cells. But really, even in the presence of that linear incision with nanoparticles down into the dermis, there wasn't a significant increase in this inflammatory response. So that was kind of interesting for us that at least from this study, this zinc oxide did not cause this systemic inflammatory response. So that's one application of this. The other is that some of the imaging that we're able to do with this modality is to look at these immune cells more closely. And another type of cell that's present in the skin are called Langerhans cells. And these are really interesting dendritic cells that look like this. These were found all over the wound. And their role is to search for antigens. So to have this dendritic arbor network to be waiting there for any type of foreign antigens to come in. And when they detect those, they actually activate. And so they will withdraw their dendritic arbor and become more round like this, and then shoot off and migrate to lymph nodes to elicit a more systemic immune response. And we think this is the first in vivo sequence of this activation that's occurring. And if we look over the course of 24 hours, we see initially there are all these immune cells throughout the wound, and then you can see that they start to look less branchy as they withdraw their arbors, and these two cells were the ones that are shown here. Really by 24 hours, there are not many of those left in the wound. They've all, we think, migrated now to the lymph nodes, and we're trying to subsequently look at the lymph nodes and see if they all showed up there and look at timing. So there's a lot of interesting dynamics that can take place there. Any questions about kind of some of those studies, those mouse studies already? Yeah. Okay. Yeah, the resolution. I passed over a lot of these details, so if you have questions, please ask. So typically, we're down on the scale of a few microns resolution. For OCT, this is with a high numerical aperture lens or objective, and our resolution is probably about maybe 2 to 3 microns in depth, and about 1 micron or so transverse resolution. In OCT, our axial resolution is dependent more on the bandwidth of the source, but because we had such a high NA, our confocal parameter is also very short, and so the resolution is really kind of dependent on both of those, the axial resolution. For the multi-photon approaches, because those again rely on that temporal and spatial confinement of those photons, typically that's in the maybe 600 to 800 nanometer range for resolution. Our depths of imaging with OCT, we can go down, for all of these, it's roughly about 500 microns or so. Multi-photon can go that deep as well in scattering tissue because it uses these longer wavelengths. Yeah, so each modality is kind of sampled as it should based on that resolution. But you're right, that when we ultimately combine these, and that image, those 3D images I showed of the mouse skin for the multi-photon and the OCM, you could see that there was an apparent difference of resolution. And so we have to kind of live by that fact, and we overlay them. We don't really interpolate the data between those. We keep those channels separate in that case. Yeah.
Actually, on that same line, have you tried doing any hyperspectral decompositions of the data? Do you get your separate channels to try to represent them in a different way?
So, I mean, there are independent channels, but when you say hyperspectral,
I mean, you think there's some sort of correlation between them, right? From the underlying materials and such. So in hyperspectral imaging, there's been some work on doing like low-rank tensor decompositions of data, kind of like a big PCA, but multi-dimensional. I was wondering if you tried anything like that.
One of my students, Andrew Bauer, is starting to look at some of these methods like that. But that it's a very good question because even though we're showing examples of say 2-photon or SHG, there's spectra associated with the signal we're recovering. So there is information contained just even within the spectrum. What we're showing is maybe just a composite amount of signal, but we could spectrally decompose that as well and look at some of those correlations too. So yeah, I think a lot of data. Okay, yeah.
When you're doing your zinc oxide, right? Zinc oxide and GFP, you had talked about before you make a peak and then you also broaden that peak when you're generating laser pulse. So are you putting 2 peaks in there, or are they excited at the same wavelength and then you select them?
So that dual spectrum usually has a center wavelength peak that has a lot of intensity that is used for the nonlinear process. It's really on the shoulders of a broader spectrum that it will be used primarily for OCT or OCM to give us a higher resolution for that. And we can, both of those, that spectrum is used to excite the tissue at each point of that scanned beam.
So you're not putting in extra peaks to excite your GFP or your those are just sort of
although we can tune. So we would tune the center wavelength of that laser spectrum depending on which fluorophores or targets that we want to excite.
But you can still, like that data that showed the zinc oxide and the GFP, that was all collected with the same.
And so in that case, the GFP emission and the zinc oxide luminescence, they were fairly close in wavelength. So we had to use filters to try to separate. There was some overlap of those spectra. But yeah, the goal would be to try to separate those.
For practical viewpoint, time, so this wound healing was done in a mouse. So for 64 days, treat the mouse like this one. And I'm just wondering, because of the stability of the laser source or this sample not moving, how was that?
Yeah. It's really a heroic challenge, I think, to do this, because the mouse is brought back at each day and then allowed to recover and go back to his cage and come back. He wasn't kept on the stage for 64 days, but was allowed to. Each imaging session was about 2 hours, and then the animal was allowed to recover and go back to his cage. And there were registration marks on the skin to basically give gross alignment, and then under microscopy to get us to align the animal even closer, and then capture images and then use the algorithm as the final approach to kind of co-register. Okay. Yeah.
You said that the zinc oxide nanoparticles were luminescent rather than fluorescent. I'm not sure if I'm clear on the distinction.
Yeah. So it really has to do with kind of the energy states within those molecules. In essence, it's an emission at different wavelengths. The fluorescence tends to have like a non-radiative decay element to that. The luminescence, one way to kind of think about that is it's perhaps more like the SHG, where there's a virtual state. The luminescence tends to be a fairly narrow line width emission, whereas the fluorescence sometimes can be a bit broader. So it really depends on the material properties as well. Okay, so talking about now turning to human skin, and these images are a bit old by now, but with just standard OCT, we can collect this data from human skin and see things such as sweat ducts and the epidermis and the dermis. If we use our high resolution OCM, optical coherence microscopy, with higher numerical aperture, we can start to see at the cellular level. So again, this is fairly shallow depths on human skin, in vivo human skin, the multiphoton channel and the OCM channel. And we see things such as the hair shaft here. These are keratinocytes. Again, different contrast. And this multiphoton is the two-photon autofluorescence of the basically endogenous fluorescent molecules, predominantly at these, I believe at this wavelength, probably the NAD and NADH. We can do this at different depths, and so we can look up to all the way down to what's called the DE junction. So these little round areas are these dermal papillae that stick up here into the epidermis. And so we're kind of down to this depth here, through the stratum corneum, the epidermis, and down into just starting into this dermis. And we can see, again, MPM tends to have higher resolution. You can see nuclei and cell membranes. A lot of those are lost in OCM. And we can see, but again, very different types of contrast. In this case, we've done wide field mosaics over roughly about a millimeter or so in each direction. And again, at different depths. The stratum corneum, which is these dead and dying corneal sites that are flaking off the skin. This is where a hair follicle was. And then it may be a little bit hard to see, but here there's a lot of different individual cells, and the dark areas on OCM are the nuclei. Do anybody have any idea why the nuclei are dark on OCM? Fred's smiling because I always ask this question. I don't have an answer to this yet. What do you think the nuclei are dark? And it's hard to see, but on MPM, they're somewhat dark, but there's some signal there. Has anybody heard of me scattering? So me scattering is the type of optical scattering or EM scattering that occurs when the scatterers are about the same order and size of the wavelength that's being used. So we're using about 800 nanometer wavelength light here. These nuclei are about that size. My hypothesis is that they're dark because they're forward scatterers. So me scattering is really anisotropic type forward scattering. And so they're dark because it's forward scattered light around that nuclei. It's not being backscattered back to the OCT detection. But that's still a hypothesis we're trying to work out. Anyways, we can do things such as identifying the nuclei in these 3D volumes and be able to characterize how those nuclei may change in size depending on different disease states, and track that type. Again, this is all label-free, non-invasive, in a grad student's forearm. Okay, well, other things we can use with, if we look at just the OCT or OCM, again, we get phase information with that, and we can look at that phase variance. And if we look at the histology or the anatomy, these dermal papillae that we were looking at before all have little capillary loops up into each one of those. And that's our skin's way of cooling, thermal regulation. So if we get, work out, if we get sweaty and hot, then we have to, your skin turns red because your capillaries are vasodilating, and you're getting more blood up to this region so you can cool off. If we look in cross-section, we see two little voids, and if we look at the time variance over time, we see that there's activity or movement in each of those voids, and in cross-section, we see the top of that loop. So with OCT or OCM here, we're actually seeing these capillary loops, and then we can do video acquisition, and we see activity in each of these little loops, and we see transient scatterers. And these are individual red and white blood cells that are circulating, flipping over each one of these little capillary loops. And so this is really something interesting that this is the first time we'd seen this. And I don't think anyone else had reported this. But it's also raising the idea, can we now, not invasively, do in vivo flow cytometry and measure the optical characteristics of those blood cells that are circulating through our skin? And that's something that we've thought of, but it's a challenge to be able to do. Okay, some other things about human skin, skin anatomy, is obviously we're aware of all the functions of skin, but this process of skin of this dying process, when skin cells that are originating from stem cells at this epidermis junction will go through a process of cell death. And so at the stratum corneum, that outer layer, they're basically just fragments of cells. There are no nuclei, and they slough off. And I don't know if it's a myth or what, but someone once said that the majority of the dust in your house is because of the sloughed skin cells that you've sloughed off. I don't know how true that is, but I don't know if it's an urban legend or maybe somebody has to quantitatively analyze that. But this happens all the time. And so our question is, how does that happen? There's proliferation down here. There's these dead cells up here. It takes about 30 days for this to happen. Well, there's obviously these changes, but does it look like cell death? Cell death can occur through two mechanisms: apoptosis, which is programmed cell death, really an intentional cell death, or just necrosis, where there's some injury and they just die. And so the question was, could we distinguish, what type of process is going on there? Well, we turned to FLIM for this, and FLIM, looking at this ratio, FLIM gives us a changing fluorescence lifetime that changes depending on this ratio of NAD, NADH. And we can look at these time constants, and basically the message is that the longer the time constant, the higher the metabolism, or the more active those particular cells are. And if we look at, in this case, we used human skin equivalents. So these are engineered skin that's grown with human cells, and they generally represent all the different layers: the dermis, the basement membrane. This is where those stem cells, a lot of the division takes place, and then all the way up to the stratum corneum. Wow. I have to speed up here, sorry. And so we can do imaging at each of these different layers and recognize these differences in cell structure and begin to combine this to look at our two modalities, MPM and OCM, and collect metrics based on the fluorescence, autofluorescence, and based on that scattering, and use that information in a very combinatorial way. So this cornification process, again, from day 1 to day 7, for each of these modalities, we see these changes, and we combine all this information to be able to distinguish what might be going on. So we can induce apoptosis by adding different chemicals. We can induce necrosis by adding different chemicals to these skin samples. And we see these changes in lifetime compared to a control. Okay, so in this case, this fluorescence lifetime for apoptosis increased, which kind of makes sense because it takes a lot of energy for apoptosis to occur, for the cell death. Whereas necrosis is really not an energy-driven process, and so that lifetime just kind of falls after those cells die. And we can see there are changes in that coloration over time. Now we combined the MPM signal with the OCM signal to tell us, how big are the nuclei. In apoptosis, nuclei will fragment, and you'll have these blebs off of you're shaking your head. So some people have heard of this process, and so we make use of the structural information here and combine it all, and we can see that very clearly we can distinguish apoptosis from necrosis, and in fact our apoptosis looks a lot more like control. It's very close to a control type behavior of cells because there's probably natural turnover of cell death, but very different from necrosis. And ultimately, the conclusion is that this cornification process looks a lot like apoptosis. And this is unpublished work, but something that we think is going to be an interesting way to be able to distinguish these processes using this multimodal approach. And any single modality alone won't give you this information. It just hasn't been as reliable, and many people have tried to do that. Okay, well, I just want to end with a couple more slides. One of the things that we do coming back into my group is that we have exquisite control over light and the properties of light, and can generate with different optical fibers. We can generate ultra-short pulses in the visible. We can generate supercontinuum. We can generate short pulses in the near infrared, all depending on how we pump optical fibers, and then use pulse shaping to control the spectral properties of the light. And we can generate, again, many different forms of light for these types of imaging and diagnostics. Well, one way to do this is to use those sources for a technique that we call NIVI, nonlinear interferometric vibrational imaging. And this is a form of interferometric CARS. So how many people have heard of coherent anti-Stokes Raman scattering, or CARS? Great, yeah, so maybe about half of you or so. So for the other half, the way CARS works is every molecular bond has vibrational states. And if you put in optical energy, two different beams that have a frequency difference that corresponds to that vibrational frequency, you'll resonantly enhance that vibration. And that vibration itself will then generate a new beam of light, the CARS beam, that's coherent with the pump and the Stokes that are driving that vibration. So it's kind of like pushing someone on a swing. If you push them at the right frequency, right, they're going to go higher and higher. Otherwise, they're going to stop. And so that's what we do. We have a pump and Stokes beam that goes in and excites molecules in tissue sections. And then we use an interferometer for many reasons, to get phase information, to improve our signal to noise, to reject the background of non-resonant signal. And we can do things such as generate now molecular maps of tissue based on the endogenous molecular vibrations that are present. And this is skin tissue from a pig, where we color-coded different vibrational states depending on the molecule that may be vibrating. And now we can generate images like this, where in this case the pinkish are more the lipids from the fat cells, the blue is more the proteins from the collagen and the stroma. And so again, label-free molecular imaging. And this is examples again of a hair follicle where it's surrounded by fat cells. We see the white is the fat cells. Here the stroma, collagen stroma is green. And we can look at assigning different vibrational spectra to different molecular features within these cells and tissues. And that gives us really a lot of interesting possibilities for this type of molecular imaging. These, some more unpublished data, but I just had to show these because these are really fascinating. This is a second integrated microscope that combines now our CARS and NIVI type imaging, the molecular imaging, with 2-photon fluorescence, SHG, now 3-photon fluorescence, and third harmonic generation. So you can go into higher orders here, weaker signals, but new information. And this is looking at breast cancer tissue. And we can see that between normal breast tissue and tumor, there's distinct differences in structure and different features. See, even in this third 3-photon fluorescence, we see different features. So these are just a whole sequence of wide field images that were taken from a study that's now ongoing, looking at the progression of tumor within a rat mammary gland and trying to get at what are the early molecular changes that take place in tumor genesis. So the scale here is, I'm going to flash through different images, but this is the imaging modality. This is CARS tuned to the protein band, and we can see a lot of the protein signatures here. These are fat cells that have low signal. This experimental means it's a tumor-bearing animal, and it's at week 7. If we look at the CARS, the lipid, we see that, and I'll flip back and forth, you can see they're somewhat complementary. So the lipids, of course, are the bright yellow in the fat cells. And then we look at, again, this is the same region, but now this is 2-photon fluorescence, and we see structures that may be ducts or vasculature. I bet they're ducts because some are filled, which is where the tumor originates. Some are still patent and open here. And again, if we flip back and forth, very different contrast. And then the SHG, which is the collagen, again, which is can be overlaid. The 3-photon fluorescence shows really interesting punctate type features in these ducts. We don't know what that is yet, but something that's very unique. And then this is the third harmonic generation, which again is going after more of the structural interface type features. You see the cell membranes around these adipocytes, and not the fat, the lipids that are inside of them. And then this is now the control. So this is a normal mammary tissue. Again, the protein here, the lipids, of course, nice distinction. The 2-photon, the SHG, the 3-photon, and the third harmonic, and a lot of interesting types of features that we're starting to see. So again, I think this combinatorial approach gives us a wealth of information. Now, the last thing then is elastography. So we can use the application of mechanical energy to drive and move tissue, and we can then use optics to measure how is that tissue moving and how are mechanical waves such as these that are shown here in phantoms of different stiffness, gel stiffness, how that changes the wavelength of those shear waves propagating through the phantoms of the tissue. We can do this to look at characterizing the elastic modulus of different areas of skin in human skin. So differences between the volar or dorsal forearm or the palm. Also differences that may correspond to the collagen, underlying collagen structures within deeper in the dermis. A lot of applications of this have been applied to trying to characterize tumors that are more stiff. And in a sense, this is mechanical spectroscopy. So if we have a tumor here next to fat cells, then this is the OCT But if we do elastography at a lower frequency, the less stiff area is going to generate and have signal. Whereas at higher frequencies of modulation, the stiffer tissue is going to produce the signal. So we can use this to differentiate the mechanical properties. I think I'll skip this and just kind of end and say that, you know, we have a long list of multimodal approaches, optical imaging approaches for looking at tissue and cells and molecules. And I've gone through all of these today very quickly. But in the application of imaging skin, they give us just a wealth of different contrast of the collagen, the metabolism, the structures, the function, the physiology. And this can all be used to look at these processes that are occurring in the skin. Not just only skin, but anywhere where we can get this light. If we can get it to the, somewhere in vivo, if we can do this in surgery with exposed tissue, wherever we can get light, we have a wealth of tools that we can use. So I just want to acknowledge my research group. Really a great group of grad students, undergrads, postdocs, and scientists, all from many different backgrounds that come together, which is really important for this type of interdisciplinary work that we do. A lot of clinical collaborators that we work with, and just a great group of people. This is our website, so if people want to see other things that we do, you're welcome to visit that. And certainly I'm here, this week and next week, and can always answer questions. And you'll have a chance to see some of the OCT instrumentation in the labs as well. So sorry I had to go so fast, but I'm happy to answer a couple more questions if you have any. Yeah.
When you do your elastography, you should consider any material you consider
So Kelvin Boyd model is really simple.
So are you also able to so you get like this
Yeah, so you see, we have different techniques where you can apply like a step response. So you have a mechanical force that maybe is applied in step or impulse, and you can look at particularly the creep effect. So it will be the slow transient as the fluids redistribute. You could also have mechanical stimuli, and so you can look at phase shifts and phase delays, and from that get viscosity out of the analysis. Yeah. Correct. Yeah, so really for...
Is it possible to repeat the question?
Yeah, so
we couldn't
the
hear
question
it.
was talking about I think the properties of the light that we would need to use for OCT, and whether it's a continuous wave, or maybe a short pulse. OCT in particular relies on low coherence light, and so and I apologize I didn't have a lot of the background slides, but so with light, when you have low coherence light, you can imagine in an interferometer you have a sample, an arm, and a reference arm. And really, if what we rely on is reflections coming back from each of those arms, and those reflections have to travel pathways that are matched within the coherence of the light source. So if you have, in essence, you can think of this as pulses, optical pulses that will travel those paths, and they have to come back and interfere in space and time. So generally, if you need low coherence light, that means an ultra-short laser pulse that's short in the time domain but has broad spectrum in the frequency of wave. A continuous wave laser, though, tends to be have a very long coherence length. And when you have that, you can't localize where reflections are coming from. Because again, you kind of have to have a very short kind of coherence window.
You talked about a human skin cancer model, and I was wondering if you could just, what substrate did you use to make that, or did you let the cells differentiate into the different levels, or see them?
Yeah, there's a couple of ways to do this. You could use some new mice, which are immunocompromised. You can actually take human melanoma cells and inject them into the skin, and you'll have a human tumor growing on that mouse. Some of the engineered tissue scaffolds can also be grown with human cells and human melanoma cells as well. Those generally, I think, are the two.
That was ex vivo, right?
Yeah. So the engineered human skin I showed, that was just normal skin cells. That wasn't melanoma.
Right.
But that we actually got that from a company that makes human skin clones. They can be grown, but it's a challenge too. Matek, M-A-T-E-K, I believe is the manufacturer of those. Yeah.
And what is the maximum penetration for
Yeah. So in skin with multiphoton, we are imaging down to about 300 to 400 microns, and then one of the questions that always bothers us too, that we haven't solved, is that when we use OCM, which is optical coherence microscopy, we get about the same imaging depth as multiphoton. Now, if we use OCT has a longer depth of field, has less, has worse transverse resolution because we have a long confocal diameter. But we can image several millimeters deep into highly scattering tissue. So why is that different? And so partly it's because with a higher numerical aperture, we have basically a larger collection aperture, tighter focus, and so are we more susceptible to collecting multiply scattered light with that larger collection aperture? And so is it the multiply scattered light that limits our depth, or is it aberrations? Because when we have a larger collection aperture, higher NA, we're more susceptible to aberrations, optical aberration. And is that worsening our resolution? So that's the end of that. So that's an unanswered question. We haven't really noticed a significant difference. And again, we're working in the near-infrared wavelengths. And melanin is really mostly absorbing in the visible wavelengths. So that's probably why we don't see it. Okay. All right. Well, thank you. Well, one more?
Yeah, I'd like to know what is the additional information that the 3-photon fluorescence can provide compared to the 2-photon microscopic.
Yeah. So that some of we see differences in the contrast, and for us, we're still trying to figure out what those differences are. So for 3-photon versus 2-photon, we're exciting different endogenous fluorescent molecules. We may with 3-photon be exciting flavins and tryptophan. These are others that absorb more strongly in the UV. So with 3-photon, we get down to the excitation. If we're pumping at 900, we're down at 300 roughly. So we're exciting different molecules there. For the third harmonic versus second harmonic, it's really based on different structures that are present, the ultrastructures present in the tissue. So it's different contrast, but we don't really know what it's.
But I think the power used for the 3-photon excitation might be very high to excite the tissue and answer
Yeah. We don't. For those images, we didn't increase the power. I don't believe so between the 2-photon and the 3-photon. So that was all with the same type of pump power. And most, I think, all those levels we were using were safe as well. We have just a real sensitive design. Okay, I think there's a couple more announcements then.